Welcome to the deep dive.
Great to be here.
Today we've got, well, really different sources lined up. It's quite a contrast.
Yeah, definitely. We have a scientific paper on nanotech for sustainability.
Right. Very cutting edge.
And then something completely different. A very personal biographical piece, originally in Persian. A life story.
So our mission today for you listening is to really dig into both. Find the key takeaways, those interesting nuggets.
And see what kind of insights, maybe unexpected ones, emerge when we put them side by side.
Exactly. Let's jump right in.
Okay.
I mean, it's striking just how different they are, isn't it? One's about microscopic science.
And the other is this really human lived experience.
Yet, maybe there's a common thread. Both seem to touch on transformation, maybe resilience in different ways.
That's an interesting thought. Let's see.
Okay, so let's tackle source one first, the scientific paper.
Right. This one's all about what it calls sustainable bioprocessing, a really promising field.
And the core idea, as I understand it, is using enzymes, those biological workhorses.
Exactly, catalysts. But the key is attaching them, immobilizing them onto really tiny nanoparticles.
Nanomaterials, yeah. Why do that? What's the goal according to the paper?
Well, the main reasons are about improving the enzymes, making them more stable, last longer, perform better catalytically, and crucially, making them reusable. That's a big one.
Okay, reusability. And why boost enzymes like this? What are they working on?
They're working on converting biomass, natural materials that can be tough to break down.
Like what specifically?
The paper mentions lignocellulosic stuff, basically plant matter, wood, crop leftovers.
Got it.
And also chitin-based materials. Think shrimp shells, insect skeletons, that kind of thing.
Okay. So taking that stuff.
And turning it into valuable products, things we can actually use.
Like biofuels.
Biofuels, yeah.
Yeah.
Also bioplastics and what they call platform chemicals. Basically building blocks for other chemicals, turning waste into value.
That's the dream, isn't it? Now, this paper, it gets pretty specific, right, about the kinds of these nano things.
Oh, yeah, it does. It details several types of these nanostructured biocatalysts that are developing.
Okay, so what's the first category?
One major type they discuss are mesoporous silica-based enzyme carriers.
Okay, silica. Like sand, but structured.
Sort of, yeah. Think tiny silica particles, but riddled with pores, like microscopic sponges. They call them mesoporous silica nanoparticles, or MSNs.
Sponges.
Key things about them: huge surface area inside, pores you can tune to different sizes, and they're great for loading up with enzymes.
So they can hold a lot of enzyme.
Exactly. The paper mentions specific types like SBA15 and MCM41, just names for particular structures.
Right. And what's a key job for these silica carriers?
Well, one application they highlight is immobilizing an enzyme called cellulase.
Cellulase. That breaks down cellulose, right? Plant fiber.
Precisely. It chops up the cellulose into simple sugars.
Sugars you can then use for
fermentation into bioethanol, for example.
Yeah.
That's a major focus, getting fuel from plant waste efficiently.
And the benefit of sticking the cellulase onto these silica sponges?
Protection. The source really emphasizes this. It shields the enzyme.
From what?
From things like high temperatures or harsh chemicals that would normally destroy it, denature it.
Okay.
But the porous structure still lets the cellulose get to the enzyme. So it's protected, but still active, like giving it a little armored suit to work in.
A tiny armored suit. I like that. Okay, so that's silica. What's next?
The second type they talk about are metal organic frameworks, MOFs.
MOFs. Different structure.
Completely different. These are crystalline. Imagine tiny scaffolding made of metal ions or nodes connected by organic molecule linkers. Creates these intricate, porous, cage-like structures.
Cages. Okay. And what do they do for enzymes?
The paper points to their ability to stabilize enzymes, hold them securely, and maybe even control when they're released.
Interesting. Any examples?
Yep, it gives a couple. One is ZIF8. That's a zinc-based MOF, apparently quite stable.
And what enzyme goes in the ZIF8 cage?
They use it to encapsulate an enzyme called butyl glucosidase.
Okay. What's its job?
It helps finish the job of breaking down plant matter, converting certain leftover sugars, oligosaccharides, into plain glucose.
Right. So sticking it in this ZIF8 MOF.
The paper says it stops the enzyme from just washing away, it boosts the reaction speed, and it helps the whole system withstand tougher conditions. So very effective little cage.
Got it. Another MOF example.
Yes, ML101CR. This one's chromium-based.
And its application?
Used with chitinase.
The enzyme for chitin from the shell.
Exactly. Breaking down chitin into something called N-acetylglucosamine.
And why is that important?
The source flags it as a key precursor, a building block, for making bioplastics.
Okay, so another route to bioplastics. And the MOF helps how?
Big time on recyclability and lifespan. It lets you reuse the chitinase enzyme way more effectively and makes it last much longer.
Okay, so we've got silica sponges, MOF cages. What's the third main type?
Magnetic nanoparticles.
MNPs.
Magnetic. Usually made of iron oxide, like phyto. And the big selling point, according to the source, easy recovery.
How so?
Because they're magnetic. You can just use an external magnet to pull the nanoparticles with the enzymes attached right out of the reaction mix.
Oh, that's clever. Practical.
Very practical. It makes a huge difference in potentially lowering operational costs in a big industrial process. No complex separation needed.
Yeah, I can see that. Makes sense. Examples here.
They mention things like iron oxide cores with a protective silica shell around them.
So combining materials.
Yeah, or magnetic nanoparticles coated with chitosan, which is derived from chitin itself.
Okay. And enzymes they carry.
They talk about using them for xylanase. That enzyme tackles hemicellulose, another part of the plant cell wall, breaks it down into xylose.
Xylose? Is that also useful?
Yep. The source notes xylose is another precursor for bioplastics. So multiple pathways opening up.
And the magnetic part means?
Easy recycling of the xylanase. You pull it out with a magnet, wash it, put it back in for the next batch. Maintains activity over many cycles.
Efficient. Any other magnetic examples?
They also mention using magnetic nanoparticles for lacase.
Lacase. What does that do?
It's good at degrading lignin. That's
Sure.
the really tough, woody part of plants.
And breaking down lignin gives you...
Useful aromatic platform chemicals, phenolic compounds, things like that. Valorizing lignin is a big challenge in biorefining.
So the magnetic carrier helps the lacase.
Increases its stability, makes it last longer, while still keeping that super easy magnetic recovery.
Right. Okay, stepping back a bit from the specific materials, what's the big picture here? What are the key advantages the paper is pushing?
Okay, summarizing the main points. First, enhanced enzyme stability. These nanocarriers act like shields.
Protecting from heat, pH changes, inhibitors,
Uh-huh.
things that usually kill enzyme activity.
Exactly. Second, higher efficiency. You can often load more enzyme onto these carriers, and the setup can lead to faster reactions. Better conversion of biomass to product.
Makes sense.
Third, reusability and cost reduction, especially of the magnetic ones and also the MOFs in some cases.
Yeah.
Being able to recover and reuse the expensive enzymes is huge economically.
Absolutely.
And fourth, the overall sustainability angle. It's all geared towards enabling the use of waste, agricultural waste, food processing waste, turning it into valuable bio-based products instead of just landfill.
So specific industries this could impact, the paper mentions.
Biofuel production, definitely.
Yeah.
Like the cellulase on silica for bioethanol.
Uh-huh.
Bioplastic synthesis using chitinase on MOFs or xylanase on magnetic particles to get those precursors like
Right.
chitosan or xylose.
Right, the building block.
And biochemical production, like using lacase on magnetic particles to get valuable chemicals from lignin waste.
It really sounds like this kind of nanotech could seriously advance what the paper calls the bioeconomy, doesn't it?
Yeah, by making these conversions.
Right.
Well, just much more practical, more efficient, cheaper, more sustainable.
Taking stuff we throw away and unlocking its chemical potential, basically, using these tiny engineered particles.
Exactly. It moves it from a nice idea to something potentially scalable and industrially relevant.
Okay.